Hello, my name's Professor Deborah Tweddle. I'm a Professor of Paediatric Oncology at Newcastle University and an Honorary Consultant in Paediatric and Adolescent Oncology at the Great North Children's Hospital in Newcastle. It's my pleasure today to introduce to you the work that we've been doing looking at optical genome mapping in neuroblastoma. And thank you for inviting me. Neuroblastoma is the commonest extracranial paediatric solid tumour of childhood. It arises from cells that go on to form the sympathetic nervous system, and so as a result can run anywhere, occur between the neck and the groin, but the commonest site is the adrenal medulla. It's the commonest tumour of childhood and the commonest tumour of babies of any type, and second only to brain tumours otherwise. However, it's one of the hardest childhood cancers to cure, but thankfully over the last 50 years or so, we've seen an increase in outcome and improvement in survival to around 65% of all children surviving neuroblastoma. We usually divide neuroblastoma into 3 risk groups, which are shown here and are low, intermediate and high. You can see on this survival curve that patients that have low and intermediate risk neuroblastoma have a very favourable outcome, and these tend to be children who are young, under the age of 18 months, have localised disease, i.e. disease that hasn't spread, and don't have amplification of an oncogene called MYC-N. The remaining cases, of which over 50% of neuroblastomas present like this, are either over the age of 18 months and metastatic, or they have amplification of the MYC-N oncogene. And you can see that these patients have a relatively unfavourable outcome, with only 50% surviving at 5 years. And it's these patients that we really need to develop new molecular tests and new treatments to try and improve their outcome. Within each country where neuroblastoma is treated, there is usually a national diagnostic service so that standardised, quality assured, robust molecular testing can be carried out in order to assign patients to the correct risk group, because they determine treatment. Within the UK, the Molecular Diagnostic Service nationally is represented in Newcastle, and here shows you the tests that we carry out here as part of the Molecular Diagnostic Service. We begin with using fluorescent in situ hybridization to look at MYC-N amplification, and if that's present, a patient automatically has high-risk disease. And we then extract DNA to look at copy number abnormalities, and you can see here that the patient has got MYC-N amplification, but also another gene that's important in neuroblastoma, the oncogene ALK. Because ALK has such an important role in neuroblastoma now, we look for other methods of testing it, including a mass spec array and also targeted sequencing, and then other ways in which we found now neuroblastoma can be classified in terms of outcome is looking at methods of telomere maintenance, and here we would use a TERT FISH technique for such detection. And then finally, now within the UK, we're pleased to be able to offer on the NHS for all children and young people with cancer whole genome sequencing. So I want to elaborate a little bit more about the other common molecular abnormalities in neuroblastoma, and neuroblastoma particularly is a copy number driven disease. And so when we undertake the SNP arrays in patients having done the MYC-N FISH, we will be looking to see whether they have copy number abnormalities that are either whole chromosomal abnormalities or segmental chromosomal abnormalities. And classically, we would use a single nucleotide polymorphism array, a SNP array, to pick these up. So you can see here in the top profile, this is what's called a numerical profile, where there are whole chromosome changes in the numbers of the chromosomes, and this is associated with ploidy and classically a low risk type of infant neuroblastoma. And when we see this kind of profile, we can be reassuring to families that either the tumour will potentially go away by itself or just mature to a benign type of neuroblastoma. However, we more commonly see the segmental type, where instead of whole chromosome changes, we see parts of chromosomes, and there are several different chromosomes where we will see what we call typical segmental chromosomal abnormalities, which we know to be associated with a more unfavourable prognosis. And here you can see a plot showing 3p deletion, 11q deletion, and 17q gain in a patient with a segmental profile. And if patients have a segmental profile as opposed to a numerical profile, they have increased intensity of treatment, and usually that will, in a patient with low risk neuroblastoma, lead to a favourable outcome. So for this study, we wanted to investigate the role of optical genome mapping to detect both the known genetic abnormalities in neuroblastoma, but also to see if we could pick up particularly structural variants and balanced translocations, which you can't see using the current methods of detection. And you'll have heard about this already, but essentially we were very lucky. We had a Sapphire instrument on loan within the Newcastle Genetics Laboratory, and we were able to identify neuroblastoma tumours and cell lines, which we could run using the Bionano profile, involving isolation of high molecular weight DNA, labelling, transferring into the Sapphire for scanning, and linearizing the DNA, and then being able to visualize the structural variants using the Access software. And so I'm going to show you data that we've done both on cell lines using the de novo pipeline and on tumours using the rare variant pipeline. So first of all, the cell lines. As I've mentioned, MYC-N amplification is a very important abnormality in neuroblastoma. So we divide tumours and cell lines on the basis of the presence of MYC-N amplification. And from this carrier type of the NB1691 cell line, you can see that there are homogeneously staining regions representing areas of amplification on this carrier type, which my colleague Nick Bound has gone on to do metaphase fluorescent in situ hybridization, showing that within chromosome 3 there is these homogeneously staining regions that represent areas of MYC-N and MDM2 amplification in neuroblastoma. And this was found alongside various other abnormalities when we first received this cell line many years ago from Clinton Stewart at St Jude's Children's Hospital, and were able to study it for our neuroblastoma research. We then compared a SNP array on the neuroblastoma NB1691 cell line with the whole genome plot that we obtained from the optical genome mapping, and you can see, comparing the two, they're very concordant, and of particular interest is the presence of these amplified genes, which we saw, one of which was MYC-N here on chromosome 2p, but also we've got another important gene on 2p, which I showed you earlier, ALP, and also on chromosome 12q, we've got a number of oncogenes which included MDM2, which we could see on the metaphase fish, but also CDK4, together with many other segmental chromosomal abnormalities which correlated between the two techniques. But what was of interest for this cell line was that using the circus plot obtained by whole genome sequencing, you could see this very prominent chromoplexy of balanced translocations between chromosome 2 and chromosome 12, indicating that there was multiple translocations. And when we looked in more detail, we found some very interesting structural rearrangements, which included a putative fusion between these two genes, PTPRR and BES3. PTPRR is a protein tyrosine phosphatase, which is involved in activating the MAP kinase pathway in neuroblastoma. And we postulated, by confirming that the fusion was present using an RNA fusion panel, that this would lead to production of a truncated protein and may lead to inactivation, therefore, of the gene and the protein, and lead to hyperstimulation of the RAS-MAPK pathway. And we postulate that this might be a novel mechanism of RAS-MAPK activation within neuroblastoma. So now turning to the SHSY5Y cell line, which is a classical non-MECAN amplified cell line, which many neuroblastoma researchers study around the world. We received this cell line from Memorial Sloan Kettering back in many years ago from June Beadler, who established the cell line from a patient with relapsed non-MECAN amplified neuroblastoma. And you can see from this karyotype that there are no homogeneously staining regions or double minute suggestive of amplified genes. But even when we did karyotyping, we found that there was presence of translocations, including a three-way translocation here between chromosomes 15, 22, and 17, but also of importance, this one between chromosomes 7 and 8, which with optical genome mapping, we were able to examine in more detail. So to initially show you the results from the whole genome view of the optical genome mapping above versus the SNP array, you can see similarly to the NB1691 cell line that these are concordant, but the difference here is that there aren't as many segmental chromosomal abnormalities as we saw with the other cell line, but we've got trisomy and of chromosome 7 and a very classical abnormality in neuroblastoma, chromosome 17q gain. When we then looked at the circus plot, we could see that these translocations that we saw on the karyotype were represented in more detail. And I particularly want to draw your attention to this one between chromosomes 7 and 8, because we have been aware of this cell line, but it's not been properly characterized until now, showing that it's between these two genes, XOC4 and PVT1. XOC4 has a super enhancer within its locus, and it had previously been shown that this was underwent a enhancer hijacking to lead to activation of MYC in neuroblastoma, so not MYC-N, but that actually led to increased MYC expression. So although not MYC amplified, there was still activation of MYC in this cell line. But what we found as a result of this work is that the fusion is with another gene that can regulate MYC, a long non-coding RNA called PVT1. And we believe that actually it's a combination of both of those with the fusion transcript confirmed by a fusion panel that's leading to increased MYC expression in this cell line, and potentially could lead to therapeutic possibilities down the line. So I now want to turn to two of the tumours that we looked at, starting with a MYC-N amplified neuroblastoma and comparing this with the SNP array with the optical genome mapping. You can see with this tumour from a patient with relapsed neuroblastoma, the characteristic peak on a SNP array and on the OGM on chromosome 2p, reflecting MYC-N amplification, but also interestingly in this tumour, the presence of additional amplicons on chromosomes 11p and 11q, which were also present on both the SNP array and the optical genome mapping. And then when we were able to look at these in more detail using the abilities of optical genome mapping to create the structural variants, you can see that there is both the two areas of chromosome 2p where the amplification of MYC-N is, and 11p and q where the amplification of the other genes are, which include other oncogenes. There's clearly chromoplexy occurring with balanced translocations, which in more detail you can see is happening here and is accounting for why we are seeing at least understanding more about how the amplifications are linked between chromosome 2 and chromosome 11. And interestingly, we can also see clearly areas of chromothripsis or hyperrearrangement, which often occurs where there are amplicons in tumours, in this case on chromosome 18. So I now want to lastly show you tumour 2, where we not only were able to compare a SNP array with optical genome mapping, but also with the recently introduced whole genome sequencing, which we're undertaking in the UK for paediatric cancers. And again, looking at the whole genome views of this time the OGM with the whole genome sequencing and the SNP array, you can see that there are lots of chromosomal gains and losses, including the hyperrearrangement and the abnormalities on chromosome 12 that are reflected in all three. But of note is that the two plots, the one for the SNP array and the optical genome mapping, are actually from slightly different parts of the tumour. And you'll see that later on when we came to look at the structural variant detailed detection using optical genome mapping and comparing that with whole genome sequencing. So here on these circus plots, the chromothripsis that we'd seen, which was apparent on chromosome 12, is very clearly seen on whole genome sequencing and on the optical genome mapping. But what is reflecting, we believe, intratumoral heterogeneity is that you can also see chromothripsis on chromosome 10 for the optical genome mapping, whereas it's not present with whole genome sequencing. And the other thing that we found is that the other structural variants that you can see with whole genome sequencing weren't all present with optical genome mapping, which we believe potentially may due to the tool used for whole genome sequencing, MANTA tool, not being as precise as the way in which the structural variants are detected with optical genome mapping. And finally, I just want to end showing the importance of being able to pick up single gene abnormalities. And in neuroblastoma, I've already mentioned that telomere maintenance mechanism detection is important, not only for detecting telomerase dependent telomere maintenance, but also alternate lengthening of telomeres, which can occur with ATRX abnormalities, and particularly in neuroblastoma, we see intragenic deletions rather than single nucleotide variants. And tumor 2 also shows an ATRX deletion, which is seen very clearly here. When you compare the tumor with the reference genome, you can see how the gene has been deleted. And this is consistent with what we detected by a SNP array also, but as you can see was very easy to see using the optical genome mapping. Just to add that one of my research assistants is also presenting a poster at the meeting with some more examples of optical genome mapping being used in neuroblastoma. So in conclusion, I think I've shown you that optical genome mapping was able to detect all the previous structure and copy number variations seen in neuroblastoma cell lines and tumours, but importantly identified additional novel structural variants which could have potential clinical significance. And the beauty of optical genome mapping is that it's a single test with a relatively straightforward pipeline for analysis. And now really what we want to look at in the future is to compare it with other long-read sequencing methods in neuroblastoma. So I just want to thank everybody involved in this work, particularly my colleagues at Newcastle Genetics Laboratories, Emily Whittle, Laura Weir, Gavin Cuthbert, and within the University, Ruby Barford, our colleagues at Bionano, and their various funding sources, including the Biobank for tumour samples. Thank you very much.